Our next speaker is Dave McColgan from Artifact, who'll be talking about when to customize design of unique visual tools in CDS.
Has anybody else tried not using any PowerPoint? I'm going to see if I can do it. Just for fun. So the text looks terrible. You'll just have to bear with me on that and trust me that it's the projector, not the design. I'm Dave. I'm going to tell you about 4 unique tools that we've built on LabKey server as part of the collaborative data space project. I work for a design and innovation company called Artifact locally. We're the kind of company that if you watch Portlandia, they kind of made fun of in season 1. We have a shuffleboard and 2 beer taps with rotating selection, and we spend serious time thinking about how best to brainstorm, things like that. So before Artifact, I was at a national laboratory creating information visualization systems for professional analysts. And at Artifact, we typically get hired to help people, organizations think about how to leapfrog the status quo or enable something new, a little bit less about incremental change. So that's what we're trying to do with LabKey. A long time ago, Artifact and LabKey wrote a proposal with Sharp, which is a local statistics group that has lots of process data already. And we got a shared grant from the Gates Foundation to figure out how to extract more value from completed HIV vaccine studies. We found investigators had tons of great ideas and lots of questions about those, but they don't have access. They're still mostly stuck with peer-reviewed publications to learn about each other's work, and that's slow and kind of an impoverished way of learning. Existing repositories had kind of turned into data graveyards because the data sets were separate, they were sparse, they were insufficiently annotated to let somebody else make use of them. So I'm going to skip right to a live demo, and then I'll come back to how we got there. So imagine I'm a vaccine researcher. I have my own data, but the data space might be the only way I can get self-service interactive access to other people's. And the first major thing we wanted to enable was, because I might not be familiar with all of it, learning about all the other studies, the assays, the vaccines that were being used across the field. And that helps me know what questions I even have and what can be answered. Most of these would have been done by institutions that aren't mine. And that helps me learn about more of them. There's also stuff from years ago. So nobody's an expert in everything among our users. Second, it helps me form hypotheses across all of these data that are brought together, we hope. So here I've started plotting one assay to look for patterns. It automatically filters to the subjects who have data for this assay, and the key concept is I don't query for data set and then make a view based on the data set. The idea of calling it a data space is basically I start with all the data, and I actually have to throw out actively the things that would get in the way of my question. So the visualization is not really a report. It's connected to an interactive flow. I can keep selecting and filtering down, and they're all going to be adding to the same filter set. So over on the right, you can see the first unique feature. This area over here is counting up how many unique subjects that I have, how many studies they come from, and some other things. So without this, I might just change the view that I'm looking at and not realize that I've accidentally gotten to a skewed population. These counts are supposed to be potential explanations for the relationship that you might see, but also the potential confounds that you need to be aware of that could get in the way of what you're trying to conclude. LabKey actually applied this design of sort of stacked filters and counts in other places. I think the last talk yesterday included Argos, and it uses something similar. And after our next development sprint, the counts will also include the number of time points that I have data for. You can imagine if a subject was tested more than once, I might have multiple data points for them. And one way to tease that apart is to put time down on the x-axis. And it's taken the exact same points and just spread them out according to when the samples were collected that the assay was tested on. And that brings us to the second unique feature that I wanted to share. So this timeline along the bottom maps to the time going across here. And there are multiple studies all aligned to day 0, but you can align them by other things like last vaccination. And each study gets its own row. I can hover over these little vaccine icons and find out exactly when which group was vaccinated and with what products. And I can even pop it open and see how the treatment arms are different from each other. So we thought this is critical to have right here when you're looking at the data, because people don't know all of the details of all the studies that they might look at from this sort of top-down approach. So it can really help you understand what you want to learn. So I might also want to look at a relationship from another assay. And while I'm in here, this is the third unique feature. People use the name of an assay a lot in conversation and get by just fine. I've heard several speakers talk about neutralizing antibody assay here, for example. But when it gets down to the details, the name of the assay can hide a lot of diversity in how it's run. Different target cells. Many of these differences are sort of like database table keys we found. You may need to choose multiple keys like cell type, analyte, and antigen in order to define what a unique row of subject data is. And our users aren't expert in every study and every assay and all of its flavors, so some users won't have been directly involved in any of it, actually. So to help them explore a relationship, we've tried to expose this hierarchy right here for awareness and control. These are the different elements that determine what it means to plot the main recommended magnitude from this assay. And we've made some default choices and recommendations that we think will be the most useful. And when I get my scatter plot, first of all, it doesn't look like there's a strong relationship, but that simple finding in our case would have been difficult to impossible to get before. And even though it's negative, I think a lot of the findings will be negative. It helps me save time and know not to invest anymore in this line of thinking. You'll notice our fourth unique feature has shown up. Some of the data are in the scatter plot here, and some are over in the margins. Every dot you see is from a subject who has data from both assays, but these data points don't have a match on the other assay, respectively. Often that's because the assays were run on samples that came from different time points, so they aren't directly comparable. But there are other reasons too, and the tooltip can tell you how much of the data are in those two categories and what the other reasons might be to help you figure out what you might want to do about it. A normal scatter plot might throw these out, but we thought it was really important not to oversimplify. I should know that these data exist, and then I could choose to export to my own tools and make some kind of cross-time comparison across multiple studies. So even though they're mismatched, LabKey's made it so there's still ways to interact with them, to probe for a relationship. Like one of my favorites is to make a little selection window and then move it around, and it's actually showing me highlighted points come from the same subjects. So if the subjects who have high data here also have high data on the other assay, there could still be a relationship that I might want to investigate, but it still doesn't look like that. So that's the four unique features. I hope it gives you a sense of how sometimes customization can really add value for everybody's unique needs of their users. In our case, the people who have access are at many different institutions all over the world. They have diverse data, roles, and expertise in different parts of the data. We spent time with a lot of them. That's our sort of approach, is the human-centered design approach. And their questions and ideas helped us really define what the program was going to be and make big choices. But through our other activities, talking to data managers and statisticians and prototyping with the real data, we found the important thing that led to a bunch of these unique features, which is, I was sort of talking about yesterday with some people, you know, the saying, no plan survives contact with the enemy. We've sort of found few researcher questions survive contact with the data. They're expert, and so it's intimidatingly smart people, and you think you could take their word for it, but they haven't been playing with it while they're forming that question, and that's kind of the point. The data are frequently different than they assume. Questions evolve as soon as they encounter the data, and then one answer leads to another, and today this is a very manual back and forth process with people if you're funded to get answers. So that's why we don't have a query and report model, and instead went with this sort of interactive flow. And there are other systems that enable interactive flow as well, but they can't provide the domain-specific annotation that we saw, data hierarchy, and guidance all in the context of the same interface. So that's that's why we made these features. So these are intimidatingly knowledgeable people, and we designed it because they're used to working with a slice, and this is kind of the whole pie. And we hope we've made it tasty. We're currently in beta, and we're going to launch in a couple of months. And if you catch me around, love to talk about the ways in which you can tell whether you need something beyond what comes out of the box and when it makes sense to invest. I think I don't have specific advice. It's just really interesting to talk about how you know and what methods you can use to find out. So that's it. I have 2 minutes left. I don't know, are we doing questions? Or? Okay, thank you.
Thanks, Dave.